Hi, my name is MNemeth, and I'm doing a study on chemopreventive foods on the allele hopscotch tumor lethal and Drosophila melanogaster. The reason these alleles form tumors on the outside of Drosophila, and they're little black tumors. Some background information: the tumor formation, they're caused because a cell uncontrollably starts over-proliferating cells, which form a mass of cancer cells. Now, the chemopreventive foods, I chose 5, are avocado, grapes, green tea, garlic, and rosemary, which all have displayed in separate studies to have anti-prolific effects on cancer cells. Now, the hopscotch gene that I'm looking at is part of the JAK family. The JAK family is unique because it has a second domain that is a degenerate kinase-like domain with that shows no catalytic activity whatsoever. The hopscotch tumor lethal allele by itself is a X-linked dominant mutation, and it happens because there is a single substitution at 341 from a glycine to a glutamate acid. The reason why I chose Drosophila as my model organism is because they're easy to attain, they're cheap, and they reproduce quickly. So my hypothesis on this study is that the 5 chemopreventive foods that I've chosen will reduce tumor size and the number of tumors seen. Now the picture here displays what a normal Drosophila looks like, but when they have these tumors, they're little black dots around the wing area. In order to obtain my hypothesis, my methods include the Drosophilus diet, which will include 6 different groups. Each one will be a different chemopreventive food mixed 50-50 with regular Drosophila food. Then I'll have a 6th control, which is just Drosophila food. Every single day I will be transferring this Drosophila to new containers so that I have fresh food. My goal is to obtain 3,000 male Drosophila since the mutation is X-linked dominant. That is 500 per group. Also, if time permits, I would also like to look at the red blood cells because this tumor does show defective blood cell shapes. My timeline begins in January when we get back from break. I hope to spend 6 weeks in the lab transferring the food and looking and collecting male specimen. And by February 15th, I hope to have this portion finished, and if time permits, to look at the red blood cells. And then March and April, I would like to have my paper written and completed. My supplies are over budget, but that is because some of the supplies are going to go to other students who are doing similar projects. My conclusion is that if I'm able to reduce the tumor size and the number of tumors using to decrease, then I'll be able to display which foods showed the best results and explain why they occurred. Oh, thank you.